Ahmed, how are you?
Afternoon, Prof. I'm good. I'm good. How's everybody doing? It's very cold here in South Africa. We've started our winter, so I'm sure you guys are heating up.
Oh,
We are going into our cold season, but otherwise,
yeah,
it's lovely. It's lovely.
Sorry. I'm
that's
sorry about
good.
that.
No, no need to
Yeah,
apologize. It's good. We get some cold weather also. It's good.
it's good. All right, friends. Thank you guys that has been here. Like we have a bi-monthly program where that leads to the journal club. For this session, we're focusing on oral microbiome. As you do know, for the early career research program run by AFRONE, it's very important for us to know critical gap areas, research areas in Africa. Like we noted, for the design of this program, we identified three critical gap areas. For one, included the use of AI and then telehealth as a critical area. And the last 3 months, we spent a lot of time around that. This is our second focus area, which is oral microbiome, studies around oral microbiome. There's very little from the African continent. And we're hoping over the next 2 months, we will ground ourselves in at least understanding the basics in a way that we can support others to
Yeah.
learn about what research around oral microbiome is like, what shouldn't make it impossible to invest that time in this research. It is my pleasure to welcome a friend and an expert in the field, much more also. She's one of the few people in Africa, I say that with modesty, not that I know any other person, that works around oral microbiome um, and oral health and microbiome. Dupe Coker is very well known in the field. She is in diaspora. She works in the US. She's going to introduce herself, but she's very much African in art, African in attitude, and Africa in prospect. Our research project is also situated within Africa, despite the fact that she works in the US. So without much ado, let me welcome Dupe Coker, who will take us through what promises to be an exciting session. Please note that if you would like to ask any question during the presentation, feel free to raise your electronic hand and I'll stop and call you. But otherwise, once she ends, we'll also take all the questions together. Over to you, Dupe, and thanks for
Thank
coming.
you. Thank you. This is such a pleasure, Ma. I don't take it. When you tell me to jump, I say how high. So thank you so much. Hello, everybody. Such a pleasure to be here. My name is Dupe Coker. I'm going to share my slides in a minute, but I talk fast. So please, like Prof said, please just flag her and she would interrupt me, or even unmute yourself. I think this was small enough to have an engaging session. I like interaction because I know enough to be dangerous. I tell people I know enough to be dangerous. I'm still learning. I'm still on a journey like all of you. So I want to be challenged. I need questions to challenge my thinking as well. So my name is Dupe Coker. I am an associate professor in the School of Penn Dental Medicine in University of Pennsylvania. I also serve as an assistant dean of clinical and translational research. What does that mean? I support students who want to do clinical and translational research. And I also I direct a center called the Center for Clinical and Translational Research here at the Penn Dental Medicine. So these are three jobs, right? But my main job as a lab PI is to, as you heard from Prof, to kind of elucidate or untangle, as it were, how factors that we know could influence the oral microbiome, and of course, understand how oral microbiome can actually influence other outcomes. So think of it like a mediating factor. Oral microbiome is a mediating factor that can affect either the relationship between something and another thing, but the mediator is the microbiome. So I set the stage for you because as I share my screen, I want to get your thinking kind of very aligned with my thinking. See if I can share my screen. Let me know if you can see.
Yes, it's coming up.
Okay, very good.
Yes.
All right. So
Perfect.
I am a dentist by training. I gave you that preamble of my current role, but I came from, like she said, a very like all of us. Well, I'm an African trained dentist, and also I have an MPH in epidemiology and biostatistics, and then a PhD in epidemiology. So it kind of tells you a little bit about why I think a little bit differently. from the average clinician, because I have to think about what does that mean? And I love this title. This title really is akin to my heart. And I was given the title by the AFRONE organizers, the oral microbiome from basics to public health implications. So this is really my bread and butter, and I'm happy to support anybody who wants to work in this field with me. Like she said, I'm one of the very few people working in Africa. There are many people doing work, but in the sense of the oral microbiome, I think I'm one of the very few. So with that, I just want to, this is what I told to always put, I don't have anything I'm going to be sharing with you that I would benefit from, no software, no commercial product, no service in this presentation. All right, so I also want to I'm going to be asking questions, and sometimes you can speak up or chat, I'll type in the chat your responses. And I want to pose you with a question. How does understanding the oral microbiome, I'll call it bacterial communities in the mouth, inform public health challenges? Think about all the public health challenges you have in the world right now. How does understanding the mouth and its bacteria inform them? Well, I will tell you, it's a big deal because the oral cavity hosts 700 plus microbial species in the mouth. And then you have about 100 million bacteria per milliliter of saliva. And of course, we swallow a liter of saliva every day, if you didn't know that. And then the vast majority, I didn't want to give a number here because people have spewed out from 20, 30, 40, 50, 60, and I couldn't figure out which one to choose. But if you read literature right now, there's a vast majority of systemic diseases that have an oral connection. And I will share some of that with you, but there's so much more that I don't even know about. Maybe you may know about it. And so it's growing in this field. So your work is important. And I want to share my agenda with you along the lines of these 5 themes. And I wouldn't do the middle one too much because the middle one, we all know about the oral disease, but you can see from the biology to the public health, I'll try and be breaking down how we kind of move from a molecular point of view to a public health point of view, right? We're zooming from cellular down to like, what does it mean for public health? Before we start, I wanted to share some basic nomenclature with you because I think this is a learning platform and I'm wanting to make sure that we all kind of use the same language. The microbiome is a collection of microorganisms and their genetic material in any given body site. So it's not just the microorganisms, it's also the genetic material, the functions, the things that the genetic function of the microorganisms or the microbes. And while the microbiota is the living community itself. It has bacteria, fungi, viruses, archaea. And then with biofilm is this structured microbial community that adheres to surfaces. And for you all that are dentists, is the basic thing is plaque, right? That very cohesive matrix that's formed on the tooth surface. And then dysbiosis is just a fancy word, which we are still trying to figure out, to be honest. Fancy word for representing the imbalance. When something is imbalanced, well, commensal bacteria or pathogenic bacteria or symbiotic bacteria, they're all not, there's a shift and it causes disease or disease progression. So these are things that I want to kind of highlight with you so you're aware of these 4 kind of nomenclature. As you read papers, as you're reviewing papers, you kind of get a sense of what you're looking at. Is it the microbiome, the microbiota, biofilm, or dysbiosis? So I want to start with what is the oral microbiome? And I know, people say, oh, bacteria in the mouth. Yeah, it is. You're right. However, you just heard me talk about microbiome is the microorganisms and the genetic material, all the functions of what they do, the expressions and the genes and the functions that they encode, not just the microorganism, but the function they encode. So it's not just saying what they are, it's expanding to what they're doing. And I want to just pause by telling you that the Human Microbiome Project, that's the HMP, they sampled 9 oral sites. And there are many more sites that are not yet sampled. And you as dentists or dental practitioners, you know more than anybody, the variability in the oral cavity. It's so complicated. I tell people that it's the most studied, but the least understood. Because one of the early microbiome studies were in the mouth. Now you see a lot of work done in the gut and in other parts of the body. But if you look back to the beginning, the early times was oral cavity. But it's hard because of the different niches that we have in the mouth, as you know more than anybody, that they have different communities. You have here plaque looking very different than buccal mucosa or hard palate. the tongue is so different, right? And so it makes our work a little bit complicated. That's why we need more of you to study it. I already mentioned this already. So you can see the oral cavity is the second most diverse or the most populated site. So you have the stool. I don't know if you can see my mouse here. Let me see if I can. The stool. The stool is the most abundant. Then you have the oral cavity, right? You have some of them coming up, the vagina, the skin on the nose, but they're still, in comparison, smaller than the stool or the oral cavity. So I will kind of start with setting the stage with how we study it. As you read papers, you would get a feel of it. Back in the day, and I wanted to give you some historical kind of context. Many of you probably were working with the latter part, or the staining, where you have culture and you stain your petri dish and you use microscopy to observe microbes. But back in the day, there was a time when there was no microscope, or there was the first time a microbe was described back in the 1600s. And then we started having a connection between microbes and disease. Then we grew into this whole desire, or the whole understanding of growing microbes in a petri dish. We culture them. But in the early 1990s, even close enough to, when I started working with this about 15 years ago, it was focused on looking at some microbes that were uncultivable. That means you can't culture, they can't grow in a petri dish. And so we started thinking about looking at DNA sequencing. We take a swab, we take a saliva, take a plaque, and we do what's called the high throughput DNA sequencing. And I'll talk to you about it a bit more, because it kind of captures much more than just microbes that can be grown in a petri dish. So with that, I wanted to kind of share with you, when I talk about the microbiome, again, not just the microorganisms, but the genes that they encode, there are many ways you can study it, right? Back in the day, like I said, we look at culture isolates. That's all we had. There was staining, institute of hybridization. But then there was an evolutionary approach, where we have think of it like a fingerprint, where you can identify microbes, either as bacteria, fungi, viruses. It's like a fingerprint, and it's called amplicon-based or marker-based sequencing, where you can look at 16S, for example, and you can actually use a section of that bacteria and identify what that bacteria is in the whole microbiome sequence that you have, because there are thousands and millions of reads, right? So you have to kind of find a way to identify which one is strep or which one is Neisseria, and that's the fingerprint that I'm talking about. So that's called amplicon-based sequencing. And what does that tell you? It tells you what is there, right? You can also look at genomic sequencing, which is whole genome sequencing, and that tells you what is there, what can they do, right? But that's still DNA based. Then you have the RNA, which is kind of driving into expression, gene expression. What are they trying to do? You know what is there, you know what they can do, but you don't know what they're doing when you focus on only DNA. So RNA will tell you that what's the activity that's happening right now, right? And then with proteins, you're looking at what is making it happen. What are the drivers for these functions? And you're also working into metabolomics, which also helps us to know what have they done, like byproducts, what has happened already. By the time you see, almost like my kids go to the kitchen and I know that somebody has come to the kitchen when I was sleeping, for example. You can see fingerprints or leftovers of things that they had done. So that's what the metabolomics is. And this is all microbiome approaches. Again, I said microbiome is the microbes and the genes that they encode, the functions, not just the microbiota, right? And so when you hear that, it's very encompassing. So I want to kind of spend some time kind of honing in on when you are about to design a study or when you're reading a paper, what are the things that you're thinking about in elucidating microbiome epidemiology? And I call that because that's the work that I do, right? Microbiome epidemiology. It's thinking about so many factors, but first of all, your question. I know there are many things on the slide, so walk with me. Is the question that you have in mind. And then that question will drive the design, because you need to know how many samples you need and how you're going to actually go ahead analyzing it, right? Then you ask a question about sample. What sample will take you to what you need, and that also goes back to I will talk to you about it because it influences the way you store the sample. For example, if you're doing a RNA, you want to make sure you preserve RNA because RNA, you don't have preservation of RNA. DNA, you can have to extract the DNA from it, but RNA, you need to make sure you have a medium like RNA later to keep the RNA active, because once it starts degrading, then metabolomics is a whole different conversation because you don't want to have a byproduct that is not relevant to the patient. So that you collect a saliva sample, you have to put in the freezer for metabolomics because you don't want it to have the byproducts that I told you about, right? Now, I talked about sample types, right? Big deal about sample types. I've talked to you about the demographics that you wanted to, almost like the confounding variables. What questions are you asking? How are you going to keep them? How are you going to collect data? How are you going to have controls, or, some way to kind of standardize your sample collection? How are you going to freeze it? What method are you going to use? I told you about many methods already. How are you going to make sure that you don't have dirty data? Quality control, bioinformatics processing. So you need to have somebody who has bioinformatics expertise. And then we dive into some really high level or exploration or analysis of the microbiome for these projects that you see on paper. So when you see a paper that has all these heat maps and they call them PCA plots, it's because of all these things that happened before, right? There's a whole lot of stuff that happened, especially this group. This G group is very important because you have to remove dirty data or noisy data from sequence data. This is a very important slide, so I can pause here if you have any questions, because I think as you think about all your analysis or your future analysis or reading papers, especially in African-based studies, I think one of my biggest things is the infrastructure to collect and store samples. And it drives everything that we do, because if you don't have good data from the A to D, then all the things that you do from E to H is really inconsequential. All right, I'll just continue because I'm not hearing anything. So I told you about microbiome, microbiota, and I kind of alluded to biofilms because I think you also hear this when you read papers. And I wanted to provide some context. So imagine you have a substrate. Either you have carbohydrates in your mouth, you eat something sugary, or some bread. I like to use the bread because it's a very clear substrate. And then microbes attached to that, right? This initial attachment can be removed with brushing and flossing, and you're okay moving forward. But then you have a time when it accumulates and it becomes an irreversible attachment. And I think there's a lot of stuff in between here that I haven't described, but you can imagine it's not a static thing, it's a dynamic thing. And then you have a microcolony formation, and you can see I put a little cover around this because it's not creating like some structure. It's like they're becoming cohesive and they're forming something that is they're speaking to each other. So they have what's called quorum sensing. One microbe is talking to another microbe and they're growing and building. And then you have another interesting way to look at it is maturation, which is think of it like a more coordinated level of microcolony, because now they are not just sensing, but they're also producing, and they're more resistant to, for example, antibiotic use because they're talking to each other. So it's like one person alone can do little, but somebody together can do much more. And that's exactly what you're seeing with they come together and they're more coordinated. So, and this is, an interesting part, because at this point in time, they're bursting at the seams and they get to disperse their contents either as a microcolony, right, or as individual planktonic organisms, right? And then you have it going all the way to pulling back to planktonic cells that come and attach eventually a substrate. So you can see this cycle is, some of these biofilms can propagate to start causing disease. Some of them break up or disperse. Right? There are different ways this can go, but this is how the biofilm cycle looks like right now that we know based on literature. I want to kind of also pause when it comes about biofilm science to kind of tell you what drives some of this. Like I showed you this whole biology of my biofilm formation, but there's so many things that influence whether it moves from, initial attachment to, the the irreversible attachment, then onto biofilm and microcolony. Like there are many things that drive some of that. And the microbiome, not just the biofilm, but the whole community and the genes that they produce is not static. It evolves across the lifespan. And so you have from birth, infancy, childhood, adolescence, adulthood, and aging. There's a whole, and I put some things there for you to think about, because there's some very keystone activities that happen in all these stages that need to be thought of as you design your study, as you read papers, and as you work with populations. Specifically, I am kind of a little biased, just like Prof. I'm biased with the children, right? I'm fascinated with the developing microbiome because it's such a unique niche, right? The factors that influence them, we know already age is a big factor, diet, dentition, and this is at the age where so many things are happening, from pregnancy, from in utero, all the way to adulthood. And we know that there are many factors like medication and disease and genetics that drive the microbiome. Again, the organisms and the genes that they encode. And in infancy, you know, the first thing you think about is, oh, there's no, they need microbes, right? They're born and, you know, they're technically sterile, but really there's a lot of like massive colonization at birth. Like as soon as they're born, so much from vaginal delivery or C-section delivery, breast milk, big, and that's where you and I work with, right, in the oral cavity. It gets to the gut eventually, but the oral cavity is the first interaction, first exposure. And we have all these very strong data that has been out there regarding bifidobacterium being a very strong catalyst in the gut and driving immune training in an early life. This is all gut, by the way. Nothing much has been done with the oral cavity. In some of my work, and I have published it, we observed a fascinating thing, and these are very young children. But we noticed that microbial diversity, which I don't think I have time to describe it today, but it's the within sample diversity, right? How many bugs are there and how abundant they are, the evenness and the richness of the bugs, the microbes in a sample. If you see here, there's something subtly happening, and this is very much mirroring what's happening with the gut microbiome. There is initially a gradual increase from, let's say, 2 years of age up onto 3 years of age, and then it plateaus, right? I didn't show you the p-values for this one, but you can see that there's a distinct difference between the under 24 months of age and 24 to 35, and then 35 to 48. There's a plateau all the way to, you know, like 5-6 years. And what that tells me is that there's a natural stabilization of the oral microbiome as soon as there's a full complement of dentition. That's why you're so important. That's why your work is so important. You as dentists and dental researchers, as soon as there's a complement that's around 3 years of age, because the diet also changes. There's a nice change in the microbiome because they now have all the teeth in the mouth, right? And so I think that also kind of makes you wonder what we should be doing as a community of dentists in researching the oral microbiome at this early age. I don't think I need to repeat myself. I think there's some very key microbes that are happening in fetus all the way to childhood. I mentioned about dentition being, for me, I've seen it strongly in my work. As soon as they have a full complement, as soon as they have at least one tooth in the mouth, the microbiome changes in the oral cavity. But from the time when they breastfeed, all up until when they're moving to adolescence, there are always these changes happening. Either the genes or the pregnancy influences, the delivery mode, the feeding mode, introduction of solid food, different things influence the way the microbiome changes over time. So we don't need to be told, on this group, you all know that the oral microbiome influences oral diseases. I don't think I need to repeat myself. Caries, periodontal disease, cancer. We kind of know this. Supragingival biofilms or the dorsal tongue biofilm, they influence a lot of what we see as dentists. So we know local. I think the systemic is what we're still growing our knowledge about. And I think even the local, I think there's a lot of understanding that we need to know about the multi-ecological factor, not just one bug. You can see here on my top here, I show you like this beautiful figure of the structure or the spatial structure of microbes as it's on the biofilm. There's like a very coherent and organized structure from strep being like outside, and then you have the corynebacterium, almost like a filamentous structure. You have a little bit of fusel kind of hiding, and fusel and Neisseria hiding in between here. So you can see they are there, but how they lay on the biofilm matters, because it's like they're building, like I told you, they're building a cohesive armor against immunity, because the job of the human body is to fight against infection, fight against these biofilms being formed, and so they're fighting against, they're learning how to fight against the immune system with this structure. The same thing with the dorsal tongue biofilm. This can happen, for example, in caries. You can see the structure of caries, and some of this could see in cancer, and even the subgingival plaque, you can see that as well for perio. So I wanted to just show you this because I think sometimes we, see if I can move this. We also forget, so we know about these two, the red complex bacteria and some of the classic cardiogenic bacteria, but there are all these other bugs that are in the mouth that also are driving disease, right? Alzheimer's disease, I'm sure you've heard about, maybe you haven't, so I can tell you, recent data showing that in the brains of Alzheimer's patients, we're seeing Porphyromonas gingivalis in the brains. So Porphyromonas gingivalis P. gingivalis is meant to be an oral microbe, right? But it's found in the brains of patients who have Alzheimer's. So they're dead post-mortem, they check their brains, they had P. gingivalis. And that has also been validated with animal studies. So that's something that we should pay attention to here. Cardiovascular disease, I think we are all trained when we're learning in dentistry how we should be mindful about those who have, some kind of cardiovascular event, and when they're having perio disease or when they're having some dental procedure being done. So I think we know about cardiovascular disease. Cystic fibrosis is also a new one. I also have some work on this, and it's hard, it's low biomass, but it's something to kind of pay attention to with lung function. Strep oralis, depending on what you're looking at, it could have an influence on the pulmonary function of these patients with cystic fibrosis. Cancer, I think this has been kind of very well documented. Head and neck cancers, esophageal cancers, or now colorectal cancers. Then we have rheumatoid arthritis as a growing field. Again, thinking about the fact that rheumatoid arthritis and perio, they have something in common. They are very immune mediated, right? So there's a commonality between perio and rheumatoid arthritis. So I understand why this would be very strong, as well as some of these other conditions. Diabetes, big deal, and pancreatic cancer. So with that, I think I don't need to sell that idea for you, but it's like we still don't know enough. And this is not just about oral microbiome. This is also for gut microbiome, lung microbiome, vaginal microbiome. There's been so much growing knowledge about the association between microbiome and disease. And this is just some of the new things coming out there in terms of gut, more gut, but I can tell you, if we want to circle each one, we'll see that there's a lot of relationships happening even with oral microbiome. And somebody might ask me, how does the microbes in the mouth, I just showed you all this data about systemic health, how does that influence, how do we get the microbes in the mouth to get to the rest of the body, or how does it impact systemic health? And there are many ways, and I just want to highlight two. You either imagine yourself biting an apple, right? Your impression of having, once you have that impact of the teeth having a very strong or firm occlusion in the mouth, there can be a blood transmission, hematogenous route, where it goes from the teeth directly to the bloodstream. Another one is enteral route, where I told you, you swallow about 1 liter of saliva every day. As I'm talking, I'm swallowing saliva. As you're sitting down, you're swallowing saliva. And that could also be through the gut. Again, microbial translocation from the mouth to the gut. And that could also drive some barrier function. So once bacteria in the mouth gets into the enteral system, it can start making the blood-brain barrier, either the brain or its different systemic levels, the function a little bit more loose. So there's opportunity for it to permeate through that blood-brain barrier. And that's what you see in IBD. right? So these are very classic examples. So people are asking, how does it matter? What does it matter? Oral cavity or oral, your mastication, it goes to the blood or through enteral route. I wanted to give you some, with all that I'm sharing with you, because I'm already halfway through, I wanted to kind of pause and tell you my own work. So I, as Prof mentioned, introduced me, I work with kids living with HIV. And back in the day, before there was antiretroviral therapy, we had these classic manifestations. Oral candidiasis, oral hericoplakia, you had Kaposi sarcoma. We knew these were classic, they call it pathognomonic signs of HIV. But with the post-heart era, we started seeing a different shift. They're all on therapy. They're doing fairly well. Of course, they have what's called chronic inflammation. But we started seeing a different kind of profile. Gingival inflammation, periodontal disease was still there, oral warts, aphthous ulcers. And what I'm going to share with you, we saw dental caries. We started observing that in this population, at least for the children, they had an increased prevalence or risk of caries. And that's work that I did. And this is, I'm really praying that all of you would kind of join me in kind of validating some of these studies, because I feel like I'm the only one seeing this. And I want to kind of expand that into other populations in South Africa, in Egypt, and other places. If you have a population of living HIV, either kids or adolescents, please reach out to Prof or I. But we started seeing, of course, clear oral pathology was higher in this population. And I'm going to clarify what these three groups are, but just to tell you that they're those who are infected, those who are exposed but uninfected, and those who are uninfected, unexposed, uninfected. And so these three groups, I studied them often, and I saw that caries was still overwhelmingly higher in this population living with HIV. And of course, they had lymphadenopathies that were higher. So I published on this a bit more beyond just in terms of clinical work. We also looked at the microbiome, and these are, they call them PCOA plots, where you can, each dot is a sample. And I use saliva, saliva samples, and you can actually orient these samples. I went through all that design considerations I shared with you from A to H. So this is the end product, but there's a lot of things that happen before that. But each sample, each dot is a sample, and I colored them based on the three groups that I just shared with you. And you can see clearly that the those living with HIV are different from those without HIV or exposed to HIV, but they are similar to those who are exposed but uninfected, right? So HI is similar to those who are exposed uninfected, but they're different from, these two are different from the unexposed, uninfected group, right? So that's an interesting finding. We found some bugs. Again, the classic strep mutans is not overwhelmingly here. We can see it's an ecological system of microbes that are differentially abundant when you look at the three groups of children. Of course, caries-free, caries-active, there was, it's not very clear here in the figure, but it's significantly different. And what was overwhelmingly for me was the breastfeeding status. So when you breastfeed for more than 6 months of age, you are very much different. You can see that it pulls away very differently from the other two groups. Never breastfed or less than 6 months. And people will say, does it mean we should stop breastfeeding? No, not even in this population, because this population is a nuanced population. Don't forget, this population is living with HIV. The mothers who are living with HIV are either transmitting their virus to their children or not. And so there's a tendency to think that those people who are breastfeeding longer are also going to be those with HIV, right? Because that's, they encourage those without HIV, those with HIV to avoid breastfeeding their children for long periods of time. Back in the day when I studied this was about 10 years, 15 years ago. So this was old data, but you can see that there's some correlation between breastfeeding and HIV status. That's why it's always good to look at the data, because this will tell you, oh, stop breastfeeding because microbiome being different, but that's not really it. It's correlated with the HIV status. All right. So all that data I showed you was really the groundwork of an NIH-funded study that I did. I partnered with Dr. Richards on, and it's really been fascinating to work with him. But what I do is kind of really drive the Nigeria part. We have a site in University of Benin Teaching Hospital in Nigeria, and we collaborate with the Institute of Human Virology Nigeria, who's like the powerhouse for getting coordinators and staff to help me do the work, because I'm in the US, right? So they have to be people that are on ground who I trust to carry out some of the work. I'm going to rush through all of this, but just to tell you that we found severity and incidence being increased, salivary flow was impacted. And one of the things you look at, this is a classic DAG diagram in epidemiology, is that there's a lot of double whammies for those living with HIV. Either the HIV or the treatment affects all these oral milieu, either the salivary flow or the tooth development, or in this case, the microbiome. And we categorize the sample, again, going back to what I told you about the design considerations. We didn't just want to call a plaque. We want to call plaque based on either if the child was caries free or caries active and what kind of lesion, either enamel or dentine. So you would know that, but the average HIV physician might not, care about it. But that's why we doing work in these multidisciplinary spaces is very important, because we can inform the way they study. They can't study HIV, just HIV, without thinking about the dental nuances to enamel, dentine, caries-free tooth, or a caries-active individual. So I think this was just kind of highlighting that there are 6 categories of caries lesions, plaque samples that you can get from, depending on whether the child is caries-free or caries-active. I'm always trying to find my mouse here. This was, I encourage you to go to the papers here, but we already have published this, and this is really exciting work. We just noticed that there was we thought that we'll see a lot of strep mutants, and we didn't see. In fact, we saw it's as if strep came and left the building. That's the way I see it, like Elvis left the building. And so it's really been a journey of like unpacking the thousands of samples I collected in Benin and transporting them to the US for sequencing and analysis. And when we look at the multi or the longitudinal part of it, we observed that there was a reduced turnover. So it's as if there's a stunted modulation of turnover with those new HIV. So it's as if they are more stable, which could be a bad thing. So in the world of adults, it may be okay to be stable. For kids, stability is a problem. It means that they are resistant to change, and change is good in promoting health because of the dynamics that I shared with you. So it was interesting to understand that what you see in adults is very different from what you see with children, right? Especially when it comes to disease and carcinogenic bacteria. All right, I wanted to also share some of these other studies that we're doing. Again, the same idea, and I'm challenging you to think about microbiome as a mediator, right? You look at one outcome and you look at another exposure. You look at exposure and an outcome, and you look at our microbiome as a mediator. So in all these things, I'm doing the same thing. I'm recycling my grants, so I'm sharing my hacks with you. So you look at HIV, which is usually the exposure, and the microbiome, and then you look at outcomes like oral HPV, and now I'm looking at cognitive function and brain. Like, so it's just like trying to understand what is irking this population, what are the things that they see in this population, and can we understand whether microbiome can be one way of doing that, one way of understanding the risk and maybe modulating it. That the goal is not just to know, is to know so that we can do better, right? Can we promote some kind of probiotic? And I will share with you some of the reasons why we should do this at the public health level. So
we have 4 more minutes to go.
Oh yeah, so thank you. So people, I already talk about the brain with you all, so I think I don't need to talk about some of the brain, reason why bacteria in the mouth can go to the brain. And some of my work is also ongoing with what's called epigenetic aging. So aging in itself is chronological or biological. Biological meaning that the body is responding to different stresses. And in this population of people living with HIV, we observed an increased risk of stressors. So they had what's called epigenetic age acceleration. And so the question was, will the microbiome modulate that? Again, my idea of being a mediator. So I wanted to kind of leave you in the last few minutes that I have to think about this public health paradigm, right? I share with you how the low-hanging fruit, you look at case reports all the way to the highest weight of evidence, which is clinical studies. But we can't do randomized clinical studies now if we don't kind of validate them in either prospective studies or with mouse models. But I'm challenging you to think about how we can grow into our weight of evidence from maybe cross-sectional studies all the way to randomized clinical trials. And like I shared with you, this is my program, basically. So I want to leave you with the public health in the last few minutes, public health implications of why we studied the oral microbiome. 3.5 billion people affected by oral disease globally. That's a lot of people. And the most prevalent condition is in the oral cavity, number 1. And 73% of this burden is in low or middle income countries. And then we have some $710 billion that dental disease carries. And this is the direct and indirect cost because of maybe loss of time at work and stuff like that. But still, it's a significant amount, right? So I challenge you to think about why you should be studying the impact of anything that influences oral disease should be studied. But why does microbiome matter? And I shared with you that if we understand that the microbiome mediates HIV, influencing all these outcomes I shared with you, that carries or HPV or cognitive function or mental health decline, then we can start thinking about different therapeutics. We can stratify our population, we can screen for them, know who is at risk for one condition or the other, stratify them, and then address some of the therapeutics for them. And I shared with you some of the dietary interventions here. One of the big things I'm looking at is what's called nitrate. Beetroot juice, for example, could be a modulatory role, play a modulatory role in modifying the microbiome and changing the risk of any of the outcomes I mentioned. And so I just want to kind of leave you with that as we think about why the microbiome can be modulated, why you need to do discovery, predictive analysis for screening and identification, and then therapeutics. And your role, and as we walk through, we need to think about how we can communicate this as we work with physicians or medical staff. For example, think about a world where you as a dentist, you can tell your patient's doctor about his glycemic control. Maybe by your treatment of periodontal disease, you can also support some treatment with HbA1c. That's the world that we should have, but right now we're not there. So finding a way to communicate this as a body to the medical team is where we have to work towards. I also want to leave you with asking, and you can take a screenshot of this, asking questions as you think about the microbiome field. Don't just say, we want to characterize our microbiome in X population compared to another population. I challenge you to think of it this way, and I give you some options here. What shifts do you expect to see? What outcome would you expect to see? What transcriptional activity for RNA, for example, or metabolic output are you looking at? Think in a very hypothesis-driven mind, because many studies I see coming out of Africa is all these global microbiome is different. What in the microbiome is different, and what does it matter? So I challenge you to ask these kind of questions. And I also want to leave you with some low-hanging fruit you can leave here with. Assess equity gaps. Who is the most in need of doing a change? Like I mentioned, my population is HIV, people living with HIV right now, but tomorrow it could be people with special needs, right? Children with special needs. Anybody that's actually not having enough support in their oral cavity. How can we also have better nutritional or microbiome interventions? I mentioned to you about beetroot juice as one modulatory role or tool. Antibiotic stewardship is one way. We can have salivary diagnostics. We can have multi-site collaborations across Africa, not just one site. And of course, medical dental integration. So those are things I'm leaving with you as low-hanging fruit. You don't have to go into the sequencing yet. Start with the things that you can do within your sphere. All right, I want to leave this again. I talked this about, I think this is easy, leaving this for you to think about how you communicate your science, how you can think about screening tools for your settings, mechanistic and causal studies, not just association studies. This is something that I deal with as well. And then validate these markers. How do we know that somebody has diabetes? Or I heard about pemphigoid being in the oral cavity first, pemphigoid in the eye being in the oral cavity first. Can you imagine that? That's amazing. So let's start beginning to think about our mouth as part of the body. I want to leave this with you. I have a lot of people to thank. I don't know how to tell you all of this. My team is not only in Nigeria, it's also in the US and Rutgers, Pennsylvania. Like there's a lot of people that I will say thank you to, because it's a village that gets to do the work. And I want to leave you with any questions you have. Thank you.
Thank you so much. Thank you so much, Duke. Thank you. This is time to ask questions. I look forward to you asking questions, but this for me is also an eye-opener. As I was following you through the presentation, a few things kept coming to my mind, but the second to the last slide kind of answered it very clearly. If we're working in the world of transdisciplinary research, oral health and microbiome study can be centered in driving that transdisciplinary research. When you're thinking of all those things, the kind of thing that comes to my mind when we're talking of changing and moving from basics to public health, is the cost implication. What does this mean when we're thinking of patient-centered care around making diagnosis based on the microbiome? What does this mean? What does this mean when we're thinking of driving research around oral microbiome and linking it to systemic diseases? But as I was going through that in my head, when I got to your last slide, I realized that it's not an option anymore. If we truly want to drive down the cost of care and improve livelihood, we have to invest in this upfront so that we can then identify what is available that is low cost for treatment, including naturopathy. I think that's what they call it, when you're looking at food that might otherwise change things, and that's low cost. But you need to understand the microbiome before you can even do that. On that note, I'll keep quiet and then open the space for questions and any other thing that would like to engage. Yes.
Thank you so much.
You're very much welcome. Mena, Khalid, Kekewa.
Thank you, Kate. That's a very thoughtful note. Thank you for saying that. I'm honored. Thank you for staying behind. And you can unmute yourself. I heard from Prof that all of you are gonna be ready to review papers on microbiome, and I'm maybe if she invites me, I'll join as well. I do think there's a lot of she's also part of a task force, African Microbiome Task Force. I do think we need more people in this field. So as you study this and you have ideas, please reach out to us. Yeah.
Thank you. Thanks for saying that. Yes, I must tell you that I think you're the only oral expert on that group. I hold that space, not because I have done anything there, but oral health must be there. And she's the only one there, So, and it's an annual meeting. So yes, if there's any curiosity, anything you're thinking about, anyone, do you want to share your thoughts, your thinking, not necessarily a question, what's going through your mind as you are listening? Or am I free to randomly call people? Okay. Adiola Williams, it's nice to have you here. Would you like to share with us anything that went through your mind? And then I'll call Professor Bayat. Adiola Williams, is there anything you'd like to share with us that you think we should take forward?
Okay, thank you very much, ma'am. Thank you so much. I'm glad to be here. Actually, when she got towards the end of the slide, because I joined a little bit late, I had another engagement in the clinic, and then she talked about looking at other population, that now the focus has been on HIV, but other populations, that got me thinking that children with special healthcare needs and all of that, it's actually an area that I think has not been well researched in our environment. And I think it's something we should look at. Thank you very much, ma'am.
Excellent, excellent. Something to think about. Yeah.
Do you have any? Does anybody see patients regular? Because there's some specialists that only people go to them because they know how to take care of people with children with special needs. Is anybody in this group like that kind of person who is specialized? Doesn't have to be a formal training, but they just have grown to be the expert in the field because people gravitate towards you for care. Anybody like that?
No, I don't see anyone.
Yeah. Okay. Yeah, that's good. So I guess what I'm saying is, as I have a population, I have a population. Go ahead. Yeah, go ahead.
Okay, so sorry, my hand was raised.
Yes.
Concerning the question, yes. So I'm not really a specialized person in the field. However, I'm affiliated to an organization that looks after children with special needs in Zambia.
Yeah.
So yeah, we must be, I think, the only organization at the moment who are tackling that. So we need some form of education in the field. Because at the moment we've just been teaching students, undergraduate students who are linked to the Copperbelt University, one of the highest learning institutions in the country. So we basically offer training in that field. However, I feel like we still need some form of tertiary training just to have some, experience and everything. But at the moment, yes, we are running in the field where we offer oral health education, treatment, and everything to do with oral health in the specialized population. Yes, so I'm here.
Yeah, I'm going to forward, Prof. There's this announcement out there for African researchers. So if you have a question like that, one of the goals of this AFRONE, I think, is to kind of help you formulate these questions. What I'm sensing is that you're looking for support for to train, but even the first thing, like, how do we care for this population? I don't think we truly know. We know what the people in Western world have told us, but I think we're different, right? I think we need to study or apply some of what they are telling us. For example, I started building on this group, and they are talking about giving the caregivers some autonomy. The caregiver can actually put some fluoride varnish, and I don't even know if fluoride varnish is common in Zambia, but I don't know if it's common, Prof, in Ife. But in the US, they are thinking about trying to empower them to apply varnish. Is that something that might be useful? I don't know. So we have so many questions that are pertaining to us that we need to study, not importing other people's ideas. Yeah.
I think that's brilliant. Thank you. That gives a segue. I think I saw Professor Bayat's hands up, and then Maha, because Professor Bayat is very much into community health, and I think we also push the issue about move care from hospital to home. Evidence in our own setting shows very clearly we're not hospital people. We only come to the hospital for curative care. So why not empower home as a source of care? So open to you, Professor Bayat. If you do, you had your hands up, and then after Maha. Yeah.
Thanks, colleagues. Thanks, Prof. It was a lovely presentation. I really appreciate it. And yeah, I was going to actually speak on a little bit other issues related to the costing. I know in resource-limited countries like ours, to do these kinds of studies are very expensive. And also, I like the slide where you say we should move away from these cross-sectional studies and just try and associate the presence of the gene with the disease. But we need to look at randomized trials or long-term cohort studies to see the actual which comes first, the egg or the chicken, because like this, we're just assuming that two microbiomes are in the place and hence one relates to the other, but where is the source and where is the metastasis? So it's important to not just look at cross-section, but then again, you need time and you need resources, and that just hinders the studies as well. So, but coming back to your second point where you say that they want to empower non-oral health personnel, and like Prof. Marnika says, that's what we're pushing for in South Africa and in many of the African countries where oral health resources are limited, extremely limited, and we need other people to actually get involved to help to deal with basic procedures like fluoride application of STF, fluoride mousse, things like this where we know will prevent the dental caries and strengthen the enamel. But again, so much of policy, so much of bureaucracy in some of our countries where it just limits, just limit whatever we want to do. But thanks for the presentation, and maybe we should meet again on out of this meeting to discuss some kind of studies that we could do. As you know, South Africa is the hub of was the hub of HIV, and we did quite a bit of research with HIV, and yeah, we could work together. I'd really like that. But
I love it.
Thanks.
I'd love to. Please, thank you. You have my email. I can put my email on the chat. So yeah, I would love to speak more.
Okay then. And Maha, we have another 4 minutes to go. Yeah.
Yeah, thank you. Thank you very much, Dupe, for the lovely presentation. Very thought-provoking. I wonder what you see as the future of microbiome research in Africa, like the 54 diverse, very rich backgrounds, countries, all of this. How do you see this? Thank you.
That's a big question. Maybe Prof, we should bring it to the task force. I see we need to form a coalition. We need to form a coalition. I think we're growing that. We need to form a coalition, but like she said, I think I'm still one of the very few people. I need more people. And that's why I love this platform, because it's growing field. And the more we get into addressing Dr. Bayat's point, getting to asking for funding that kind of feeds into large multi-site studies where we have partners in multiple countries. And maybe someone like me or someone like Prof that has a reputation can be like a lead where we can then get young investigators to think, even if it's just small studies, and sequencing has become cheaper, to be honest, right? There's a group called Inkaba in South Africa. I think they do some shotgun and 16S data as well. So I think, to answer your question in a simple way, and it's a very complex question, Maha, you gave me, but I think we need to form coalitions. We cannot do things alone. Now more than ever, because now but NIH doesn't really fund international anymore, which is my main sponsor. And I have to start thinking about, I still love my country, I still love Africa, I still want to work in Africa. So how do I keep working when my major funder doesn't want to necessarily work in Africa? So that's a hard thing. That's why we have to work together. Yeah.
Thank you so much, Modupe.
Very much agree. Very much agree. Thank you very much. Thank you.
I think just for the last 2 minutes, this is to stimulate thoughts and thinking about all of you. First of all, to remind us of what AFRONE platform is. What we've seen based on Maha and Bayat's study is that what is happening in Africa is very few, very low. And when we seemingly have high quality research, we're starting to see that is the line of North-South collaboration along the history of colonization. We're not seeing South-South. If we're going to move forward and change the paradigm of being named the poor African people, whereas we have so much resources, we have to do work. What we heard Professor Coker today is that there's still some basic works that need to be done. So much basic work. We have to ask the question and start to do those basics that will inform the bigger. Yes, we need money, but even the little we can do, we still can explore those little that trace the ground for the bigger microbiome study. So ask those questions. And indeed, Dupa is here. Do you want to engage with her? Let's continue this conversation on the platform so that we richly understand what we can do, not just because of us, but so that we can also transform the next generation of research in the space, enough of epidemiology, basic prevalence study, but more research that can impact and change the paradigm of what we're seeing in Africa. Before I shut my mouth on top of the hour, please note that 2 of you would need to present a journal next month. Please, the 2 of you, no, I think it's later this month. The 2 of you that might want to present, please shout out to me so that we can plan your session. And Professor Coker has graciously noted she will be able to join if the time, yes, is right for her. So we do have something up our sleeves. Please shout out to me if you otherwise will want to do the presentation. On that note, I want to say thank you, everybody, for being here. Let's continue our conversation on our platform. And for those not on the platform, join AFRONE. We hope to see you next year, next batch of early career researchers, and wishing you a wonderful year. Bye, everybody. Thanks for being here. Bye.
Thank you so much. Thank you. Bye-bye.
Thank you.
Thank you so much.
Bye.